The cell signaling pathways of vision and smell in humans are two extremely important senses that we are constantly using. These two pathways are vital to the survival of most multicellular organisms and have specific steps for each. The first pathway is vision. Here we have an eye with its labeled parts, and the signal transduction pathway begins when the photons of light hit the rhodopsin proteins in the rods and cones, which are G protein-linked photoreceptor cells located in the retina. Rods are receptor cells that detect black, white, and gray and are needed for peripheral vision, and cones are receptor cells that sense detail and color. Since the ligand or signaling molecule is a photon, the pathway is also known as a phototransduction pathway. After the receptor is activated through isomerization by the photon, a transduction second messenger cascade is induced. Here we have a rod receptor cell, which has membrane shelves lined with the protein rhodopsin. The G protein-coupled receptor rhodopsin converts to metarhodopsin 2, a light-activated form of rhodopsin, which activates transducin, a G protein, and GTP is converted to GDP in the process. Second, the A subunit of transducin then breaks off and activates phosphodiesterase, or PDE, which hydrolyzes cyclic GMP and then indirectly activates the second messenger camp. Because cyclic GMP becomes scarce, the Na+ channels close, and the membrane is hyperpolarized, causing the rod receptor cell to turn off. Finally, an electric impulse is then sent to the brain via the optic nerve. This electric impulse is then processed in the lateral geniculate nucleus, part of the thalamus, into an image. When the brain receives the collection of nerve signals from the eye, it interprets where the signal comes from to reconstruct the pixelate map. Retinal horizontal cells are a type of neuron that use local signaling to receive information from rods or cones and reduce the effect of surrounding rods and cones, which helps to sharpen our image by increasing the contrast. Some other neurons that process image include ganglions and amacrine cells. The axons from ganglion cells carry impulses through the optic nerve tract that terminate in the lateral geniculate nucleus.
So the second pathway is for smell, also known as olfaction, which uses chemoreceptors that receive signals and information from molecules around us and the air that we breathe in. This begins when an animal inhales volatile particles that something is giving off. This something can be anything from food, flowers, chemicals, etc. The nostrils are used like a vacuum and allow the molecules to be transported up into the nasal cavity. Although most particles are filtered out, some still make it to the olfactory epithelium, which we will talk about in a minute. Here you can see the ligands or odorants from whatever it is that the person is smelling, and they travel up through the nostrils into the nasal cavity. Once the molecules go up into the nostrils, they'll begin to explore the nasal cavity. First, they'll see the olfactory epithelium and its many different parts. They'll come across a thick layer of mucus. They'll dissolve into it and eventually reach the columnar supporting cells that shield the many olfactory sensory neurons. Here, the odorants meet with their receptors, which bind like a lock and a key. These different shapes are the reason that we smell things differently. Once the signal is matched, it's sent up the axons into the olfactory lobe or bulb. Here they gather in the glomerulus, where the info is translated into something that the brain can understand. From there, they take the mitral cell, which sends the signals along a long olfactory path into the brain or the olfactory cortex to be processed. This is a more in-depth look on transduction, the process where information is introduced into another cell. So this is between the molecules and their receptors that fit like a lock and key. So this process is a reaction to the electrical reaction created when neurons cilia bind to the odorants. So first we have the odorants that bind to the receptors. Next, the receptor activates the G protein. In this case, it is the G protein GALF, or G subscript OLF. The G protein activates adenylate cyclase, and this adenylate cyclase converts ATP into cyclic AMP. A channel is later opened that allows Na+ and Ca+ molecules inside. This whole process is known as depolarization because the membrane is becoming less negative in the cell. This is because of the positively charged molecules that are entering. As previously mentioned, the olfactory axons join with the mitral cells that further send signals to the brain with the use of the olfactory tract. The part of the brain used for this is referred to as the olfactory cortex. This later triggers a response in either the frontal lobe or somewhere in the limbic system. From there, the organism is able to react in various ways, including acknowledging or recognizing the odor and/or associating it with past memories. The brain has many different ways of responding to the stimulus.